Hi everyone. Here we are at our final lecture for chapter 3. After this chapter, we will take a detour towards the back of the book and look at chapter 24, where we're going to learn how mitochondria function. But for right now, we're going to wrap up our look at cells in chapter 3. So let's start with our not really attendance questions. To which class of macromolecules does DNA belong? What are the monomers for DNA? Where in the cell is DNA found? Which organelle makes protein? And according to cell theory, where do cells come from? So go ahead and pause your video, try to answer those before listening to the correct answer. Now hopefully you've done that, so let's see what we've got today. To which class of macromolecules does DNA belong? Well, the Na stands for nucleic acid. So DNA and all the different types of RNA are nucleic acids. What are the monomers for DNA? Remember, all nucleic acids are made from nucleotides. Where in the cell is DNA found? DNA is found within the nucleus, and remember we looked at all the different protective mechanisms that the nucleus used and DNA itself used to keep that DNA safe. Which organelle makes protein? Ribosomes make protein, regardless of if they are free floating in the cytoplasm or if they're bound to the rough endoplasmic reticulum, the RER. And according to cell theory, where do cells come from? Remember, all cells come from pre-existing cells. A cell divides, now we have 2 cells. Those 2 cells divide, now we have 4 cells. All cells come from pre-existing cells. Now, all of these are going to be important today, and you can see pretty evidently we are going to be looking a lot at DNA in much more depth and how it functions. So, depending on where you are in lab right now and when you're watching this video, you might have already spoken about this in lab. This is a big topic in lab. We also discussed it in lecture. So we are first looking at mitosis, or cell division. We're looking at that part of where do cells come from, pre-existing cells. And we can show the life cycle of a cell with this diagram that we have here called the cell cycle clock. And really this is just a way of saying the entire life of a cell from start to end, or from start of one cell to the point where it divides and now we have 2 new cells, is represented by this kind of clock-shaped diagram. Now, most of the cells in our body are not dividing. We don't need them to divide. So most of the cells in your body are not in mitosis. Instead, they're in this area right here called interphase. Interphase. And we don't need cells to divide unless we are either actively growing or if we are fixing some sort of damage. If you get a cut, well, we need to patch that cut, and the way that we do that is by making new cells. Okay, but most of the time the cells in our body are in interphase. They're not actively dividing. But interphase is actually broken up into smaller stages. So first, we will actually say most cells in our body are actually not even in one of these 3 stages that we see right here. We have something off to the side called G0 or G0. And that is when a cell is just living its life, doing its thing and not needing to go into any sort of cell division. But let's say we have a cell that needs to go into cell division. It comes out of G0 into G1, G for growth. And what happens during the G1 phase is the cell is getting bigger. It's replicating things like its cytoplasm and all the liquid and organelles found within. If we're going to make 2 new cells, we need enough contents for 2 cells. So during G1, the cell is not dividing, but it's preparing to divide. It's making all of these extra organelles and cytosol. Then we move into S phase. And S, think synthesis of DNA. If you're going to have 2 new cells, each of those cells need a complete set of DNA. So we're going to take the DNA and duplicate it. We're going to synthesize more DNA. After that, we go into G2, growth and final preparations for division. What we're doing is we have different structures in the cell that are making sure we have enough stuff to make 2 new cells. At each of these stages, we see 1 right here, but really at each of these stages, there are checkpoints. And those checkpoints are there to make sure everything that was supposed to happen in that phase did happen. We don't want to move out of G1 into S until everything that was supposed to happen at S actually happened. And then we get to S phase. We don't want to move out of S phase into G2 until we know that there is a complete duplicate set of DNA. As long as that's happened, then we move into G2. G2, we make sure everything is where it needs to be, and as long as it is, we move through the G2 checkpoint into mitosis. Now, mitosis is the point where we're going to divide the cell from 1 cell into 2. Mitosis really is the division of the nucleus and all of its genetic material, the DNA. But we sometimes just call mitosis cell division. In reality, again, it's the division of the nucleus and the DNA. And there are different stages to mitosis. Prophase is the first step. Pro means before. So prophase is the first step. It happens before all of the others. Metaphase. Meta means in the middle. We'll see what that refers to in just a moment. Anaphase. Ana means apart. And we'll see why that is in just a moment. The last part of mitosis is telophase. Telo means distant, and there's a reason for that. But during all of these phases, it shows it starting around anaphase, but really it can happen earlier than that. During mitosis, the division of the nucleus and the DNA, we also have to divide all of the rest of the cell, the cytoplasm and everything else. That is cytokinesis. And usually when we're writing these out, we show cytokinesis at the end, but really cytokinesis is happening alongside mitosis. We just happen to notice it the most near the end of anaphase when we see something called the cleavage furrow forming. Once we get to the end of mitosis and cytokinesis, we have 2 cells that are identical to the cell that we started with, just a little bit smaller. We go through a little bit of growth, a little bit of G1, and then it's a matter of, do we need more cells? If so, we keep going through this cell cycle until we have enough cells. At that point, once we have enough cells, we exit and go into G0 or G0. So that's the cell cycle clock. But now let's look a little bit more detail at the stages of mitosis. Here we have a view of an actual cell, and down at the bottom, a drawing to kind of show us what happens during each stage. A cell in interphase has DNA that is in its chromatin stage. It shows us the chromosomes here, but really in reality, that's kind of a mistake. In interphase, if you look at a cell under a microscope, it's just got this haze. You cannot see individual chromosomes. They are only showing us this because they're wanting you to see that there's ones they draw as purple and ones they draw as green. And the significance of that is to show you the purple is one set of DNA. The green is a complete duplicate of that set of DNA. So there's two of every bit of DNA. We would not see it here. We would not see it here because it's really, really hazy. It's not condensed into chromosomes yet. But if there is two of every bit of DNA, that means we are at least at the end of S phase. So we are in the interphase, but we're at least at the end of S phase because we've duplicated our DNA. We know we're in interphase because we have a very, very well-defined nucleus with hazy DNA. Here we have a very well-defined nucleus, hazy DNA. Our centrioles, we have two centrioles within a centrosome. Here we have two centrioles within a centrosome. So the centrioles and the centrosome has duplicated. Means we're getting ready to go into mitosis. Now, once we go into mitosis, remember the first step was prophase. It shows us early and late prophase. You do not need to know early versus late for our class. You only need to know prophase. They show you two different stages here because prophase can look significantly different depending on where in the phase you catch it. At first, the first thing that happens in prophase is the DNA condenses into chromosomes. So that's what we're seeing here. Up here in the image, you can start to see the same thing happening where they start to kind of look worm-like. The centrosomes start to move away from each other towards opposite ends of the cell. We say they move towards opposite poles. Here, they're starting to move away from each other. Over here in interphase, you can see they're very close together. Here, they're starting to move apart. And they start to grow these microtubule projections. Some of them move just towards the edge of the cell. Those are called the aster. That means star. Some of them reach towards each other. That's called the mitotic spindle. Later in prophase, the nucleus, the wall of the nucleus, that nuclear envelope, has completely vanished. It's dissolved. The centrosomes have reached opposite poles. They've reached the opposite ends of the cell. And the mitotic spindle is grabbing each chromosome and will be moving it towards the middle of the cell. Here, we're in metaphase. Meta middle. Everything is lined up along the middle of the cell called the metaphasic plate or the cell equator. Everything is lined up right along the middle, so metaphase. Here, not a very good example. If you looked at the onion root tip in lab, some of them were really, really in a straight line. And that mitotic spindle is holding on to all of the DNA, holding on to each of the chromosomes. Now the mitotic spindle, they're going to start shrinking. Some of them are going to start pulling towards this set of centrosome. Some of them are going to start pulling towards this set of centrosome. And when they pull, they're going to pull those chromosomes apart. Apart. So as those spindle arms begin to shrink, the chromosomes are going to be moved towards opposite ends of the cell. A complete set of DNA moves towards one end, a complete set of DNA moves towards the other end. Once they get to opposite ends of the cell, we're in telophase. Telo, distant. The chromosomes are at their most distant from each other. During this phase, the DNA, the chromosomes, are going to start to decondense. It's going to start to look hazy again. It's going to be just chromatin. The nuclear envelope is starting to reform. The nucleolus starts to reform. So that is the part that we call mitosis. At the same time, even though we don't see it happening in all those other phases, all of the organelles are moving to opposite ends of the cell also. And usually around anaphase, we start to see the inside pinching of something called the cleavage furrow. We can see it pretty evident right here, but this is the actual division of everything else in the cell. We're going to pinch this cell in two until eventually we get two brand new cells that are in interphase. They are exactly identical to each other, and they are exactly identical to the cell that we started with. That whole thing is mitosis and cytokinesis. So cell division, mitosis and cytokinesis, brings us to cancer. Cancer, we will talk about quite a bit in this class and in Bio 139. Cancer, really when it comes down to it, is just runaway mitosis. It's cell division when we do not want cell division. We said that cells only divide if we're actively growing or if we are fixing some sort of damage. But how do cells know? Cells have something called contact dependence and anchoring dependence. Cells hold on to their neighbors like we saw back in cell junctions. And when they are touching their neighbors, they know that. And if a cell is touching its neighbor, it doesn't need to make more cells because it's already touching its neighbor. Let's say you get a cut and damage some tissue. Well, cells on one side of the cut and cells on the other side of the cut are no longer touching their neighbors. So we need to fill in that gap by making more cells. As those cells divide and divide and divide and that cut closes up, well, now those cells know that they're touching their neighbors and we can turn cell division off. Anchorage dependent. Well, what about cells that are at the very, very edge of a tissue? Well, they're holding on to something like areolar tissue or something like that, something in the extracellular matrix. They know that they're anchored in place. If they're not, we need to make some cells until they are. But once they're holding on to something in their surroundings, then we turn cell division off. So anchorage dependence and contact dependence, those things turn off mitosis and cytokinesis. But sometimes those things don't work. So we get division when we shouldn't. If cells are touching their neighbors and they're anchored to their surroundings, but we keep getting cell division anyway, that's what cancer is. So why isn't every cell in our body constantly having cancer? Well, one gene called P53 is sometimes called the guardian of the cell cycle clock. Let's go back to our cell cycle clock. We have these checkpoints that we talked about. These checkpoints look to see, is this cell doing everything that it needs to? If not, do we let it go? No, we shouldn't. But sometimes we do. These checkpoints can be interfered with. P53 is the guardian of the cell cycle clock. P53 is the gene that says, yes, go ahead and go through cell division, or no, do not go through cell division. If a cell is anchorage and contact dependent, P53 should say, even though a cell is where it needs to be, do not go through cell division. Well, cancer can override that. Cancer can say, we're going to go ahead and go through cell division anyway. Something is inhibiting P53. We start to have a cell divide and make two new cells. When those two new cells divide, what do we know about them? Well, the cell that we started with and the two cells that we end with are exactly the same. If the cell that we started with was cancerous, now we have two cancerous cells, then four cancerous cells, and they keep dividing and dividing and dividing, producing more and more and more cancerous cells. So, cancer is just mitosis that's gone off the tracks. It's runaway mitosis. So let's pull out a thinking cap question. We haven't had one for a while. Why will there never be a cure for cancer? And similarly, why can we safely say that anything purporting to be a cure for cancer is a scam product? Think about that for a minute. Remember, thinking cap questions make excellent bonus questions on exams. Why will there never be a cure for cancer? Because there won't. That's a safe statement to say there will never be a cure for cancer. Yes, there may be a cure for breast cancer. There may be a cure for small cell lung cancer. There may be a cure for pancreatic cancer. There may be a million cures for every type of cancer there is. But there will never be something that is a cure for all cancer. Because cancer, what was it? It's runaway mitosis. We would have to stop mitosis to stop cancer. That's the only thing that all cancers have in common, is that they are mitosis when we shouldn't have mitosis. But every cancer is different. There's different causes. Cancer is not a thing. It's an umbrella. There's countless different types of cancer. And the only thing that all of them have in common is runaway mitosis. Well, we can't stop mitosis. We need that so that we can grow and so that we can repair damage. If you turn off mitosis, you would die really quickly. So there will never be a cure for cancer. But we might at some point have cures for all types of cancer. So in the future, it is very possible that cancer will not be a death sentence anymore. It may be eradicated, but not because there is a cure for it. It's because there are lots of cures for all the different types. Similarly, why can we say that anything purporting to be a cure for cancer is a scam product? Because you can't stop mitosis. Nothing can stop mitosis and still keep you alive. So we have to have targeted cures for each type of cancer. So now let's move away from mitosis and cell division and talk about genes. Genes is a word that we've all heard, but we might not know exactly what genes are. So what are genes? The book definition, or the most common definition of a gene, is it is a region of DNA that codes for RNA. Gene is a region of DNA that codes for RNA. But that doesn't really tell us much about what DNA does. So the definition that I like to give for DNA is DNA tells our cells which protein to make and when to make it. DNA tells our cells which protein to make and when to make it. DNA is the recipe for protein. But this is a kind of long and complicated multi-step process. And we're going to go through that process over these next few slides. And it's going to all be based on something we call the central dogma of biology. This idea is true for all living things. Remember, all living things were made of cells? Well, all living things contain DNA. Whether it be a bacteria, a mushroom, an oak tree, a whale, a dog, a human, all living things are made of cells, and all living things contain DNA. And DNA functions the same in all living things. It does that following the central dogma, which says we start with DNA, which can replicate itself like we just saw previously. But we start with DNA, and from that we make RNA. That's step one. And from RNA, ribosomes read RNA, like we said in a previous lecture, and make protein based on what that RNA says. The central dogma of biology says we start with DNA, from that we make RNA, from that we make protein. DNA to RNA to protein. What does protein do? Why is it so important? Think back to the end of chapter 2. Protein does everything. Protein is the most diverse and versatile macromolecule in our body. It does everything from give your skin its texture, to give your eyes and hair their color, to lubricate your throat, to protect you from disease. Almost everything in your body is performed by protein. Where did that protein come from? From DNA containing genes for that protein. Now, when we look at DNA, remember, DNA was made of ACTG nucleotides: adenine, cytosine, guanine, and thymine. And they are in a different order for every single living thing, unless you are identical twins or a clone. So all living things, except for those two examples, all living things have DNA written in a different order. The A's, the C's, the T's, the G's, they're all in different orders. But the order determines the protein. We're going to see how that happens in just a little bit. But we will say we're going to break the DNA into 3-letter chunks, 3-letter words, we will call them. So maybe we have ACT. Maybe another one is GCA. On and on and on. So 3 letters of DNA at a time. What do we call 3 letters of DNA? A triplet. A triplet is 3 letters of DNA that code for 3 letters of RNA. 3 letters of DNA that code for 3 letters of RNA. Now remember, if we looked at DNA and we knew what letters there were, A's and C's and G's and T's, well, we knew automatically what was on the other side. If we had an A on one side of DNA, we had a T on the other side. If we had a C on one side of DNA, we had a G on the other side. Base pairing. But from that DNA, we're going to unwrap it. We're going to take that double helix and peel one side of it away to expose one side of that DNA. We have to do that if we're going to make protein. We take that DNA and we unzip it. We expose one side of the DNA. So we have A's and C's and G's and T's that are not attached to anything. And that's good, because base pairing doesn't just hold DNA to DNA. It also allows us to duplicate DNA. That's how our DNA duplicates. It unzips itself and then it starts to stick nucleotides to those exposed areas. But that's also how we make RNA. If we have an A on one side of DNA, we have a T on the other side of DNA. If we have a C on one side of DNA, we have a G on the other side of DNA. That carries over for RNA also, with just one little difference. If we have a G on the DNA side, what are we going to stick to it to make RNA? If we have a G on the DNA side, we stick a C of RNA in place. If we have a T on the DNA side, we stick an A in the RNA space. Just exactly like making DNA to DNA, DNA to RNA is the same. Except the one difference is if we have an A on the DNA side, we're tempted to put a T for the RNA there, but remember, RNA doesn't have thymine. Instead, it has uracil. So if the DNA has an A, instead we put a U, uracil, of RNA in place. So we have this long strand of DNA, and then we build our RNA off of that. What's the point of that? Why do we need to concern ourselves with RNA? And by the way, we will come back to what I was just saying. We're going to come back to this and illustrate it. So don't worry if me just saying it all doesn't make sense. We're going to see it written out in just a moment. We have our RNA. What's the point? Well, remember, ribosomes read that RNA. And ribosomes make protein based on that RNA message. But as we mentioned in chapter 2, there's a lot of different types of RNA. We are concerned with 3 of them. 2 we're really concerned with, but 3 of them we're going to talk about in our class. There's mRNA. This is called messenger RNA. That's what we just made right here. We take the DNA, we make RNA. More specifically, we take the DNA and we make messenger RNA, mRNA. Remember, we had 3 letters of DNA that we called a triplet? Well, the matching 3 letters of messenger RNA is called a codon. 3 letters of DNA is a triplet. The 3 letters of mRNA that we build from that is called a codon. And codons are going to be used to find out which protein we make using something called the genetic code. That's what we see over here. This big, intimidating chart that no, you do not have to memorize, because just like the periodic table, anytime you need this chart, it will be given to you on an exam. You do need to know how to read the chart, but you do not have to memorize the chart. So, we have our 3 letters of DNA. We make our codon, our 3 letters of messenger RNA. We take those 3 letters of messenger RNA, we look them up on this chart here. 3 letters of messenger RNA we look up on the chart. And based on which 3 letters of messenger RNA, it tells us which amino acid that the ribosome is going to lay down. Why are we talking about amino acids? Well, what were amino acids? Amino acids were the building blocks of proteins. They were the monomers that proteins were made from. If we stick a bunch of amino acids together, eventually we're going to get a protein. So that's how the genetic code works. DNA codes for messenger RNA. We look up 3 letters of messenger RNA, codons, on this chart to see which amino acid, and then what's the next amino acid, and then what's the next amino acid, and what's the next amino acid. Eventually we have this long chain of amino acids that becomes a protein. So if we know the order of DNA, then we automatically know the order of messenger RNA. And if we know the order of messenger RNA, we will get to look up the order of amino acids. And the order of amino acids determines the protein. But we're getting ahead of ourselves. Let's back up. We know that the DNA tells us what the mRNA strand is going to look like. The mRNA is what the ribosomes read. The ribosomes are reading the mRNA and laying down amino acids. But where do those amino acids come from? How do they get to the ribosome? Well, we have another type of RNA called transfer RNA, or tRNA. These guys have 3 letters exposed called anticodons, and they are the opposite 3 letters of the codons of messenger RNA. Based on the codon of messenger RNA, the anticodon of transfer RNA is going to match with it. tRNA, what's it transferring? Well, tRNA carries amino acids. Each different tRNA carries a different amino acid. And based on the codon matching with the anticodon, the transfer RNA gives the amino acid to the ribosome. The ribosome then joins it to the growing strand of amino acids. And again, we're going to draw this out. Right now we're just walking through it with words. Last type of RNA is rRNA, or ribosomal RNA. Ribosomal RNA and protein are what ribosomes are made from. Ribosomal RNA and proteins are what ribosomes are made from. So now let's talk about how the whole process works. How are we making protein based on all of that? Well, we have 2 stages, and I'm going to back up a few slides here. This was our central dogma. We start with DNA, we make RNA. We have our RNA being read by ribosomes, we make protein. There are 2 stages. Transcription is reading DNA to make RNA. Translation is reading RNA to make protein. Transcription and translation. Step 1, step 2. Both of these steps together, transcription plus translation, is something called protein synthesis. Protein synthesis, read DNA to get RNA, read RNA to make protein. So here is the example from our book. We're going to go through this ourselves. Here is our DNA. We unzip it. We're looking just at one side of the DNA. That's what we have right here. We're going to add, if we have a T on one side, we have an A on the other. If we have an A on one side, we have a U on the RNA side. If we have a C on one side, we have a G on the other, on and on and on. Here's our DNA. Here's our messenger RNA. Here, this guy that looked kind of like a guitar, this is our transfer RNA. Here is his anticodon, and here is his anticodon. Here he is carrying an amino acid. Here is the next tRNA. With his anticodon, he's carrying a different amino acid. How do we know which amino acid each one is carrying? Well, because we have our code right here. We will be given this code. So let's go through this step by step. We have our DNA, and we know the other side of DNA because of our base pairing rules, but we don't need that other side of DNA. We're concerned with this one. We say this DNA has a known gene. We know that there is a gene in this strand of DNA. But we don't know anything else beyond that. So let's work through it and figure out what we can do here. What's the first step? Transcription. And what do we do during transcription? We're going to build our messenger RNA. See if you can do that. I've already broken it into triplets. I've put a little space there so we have our triplets. Now, using the genetic code, see if you can determine what the messenger RNA will be. I'll start with you. If we have a C on one side, down here, what do we have? A G in the messenger RNA. If we have a G up here in the DNA, we have a C down here in the RNA. So keep going all the way through. Remember what happens when you have an A on the DNA side. And see if you can get the messenger RNA, which should look like this. Here is our messenger RNA. If there's a C on one side, there's a G on the other. If there's a G on one side, there's a C on the other. If there's a G on the DNA, there's a C on the RNA. If there's a T on the DNA, there's an A on the RNA. But if there's an A on the DNA, there's a U on the RNA. So now we have our triplets of DNA, and we have our codons of messenger RNA. But here is where we run into a bit of an issue. Where's the DNA found? It's in the nucleus. We're protecting it. To protect it, we need to make sure that it stays in the nucleus. So the DNA is in the nucleus and cannot get out. But where are the guys that make protein? Where are the ribosomes? They're out in the cytoplasm, either floating around or attached to the rough endoplasmic reticulum. So the recipe for protein is in the nucleus. The ribosomes that make the protein are out in the cytoplasm outside the nucleus. The DNA can't get out. The ribosomes can't get in. So what happens? Well, the messenger RNA is going to carry the message of the DNA to the ribosomes. Messenger RNA can fit through the nuclear pores. It can leave the nucleus, and it does. And what's going to happen when it gets out into the cytoplasm? A ribosome is going to attach to the end of it. It's going to attach right here. Now, if this were a real gene, this would be hundreds or even thousands of codons long. I've just shortened it for ease of our class. But the ribosome is going to attach right here. And it's going to see GCG. It's going to read that first codon, and it's not going to do anything at all. It's going to just move on, and it's going to read CGU, and it's not going to do anything at all. It's going to keep reading codon after codon after codon, and it's not going to do anything at all until we get to the magic codon that says AUG. Here is our genetic code. All of these have a 3-letter RNA, a codon, and 3-letter abbreviation for an amino acid. But this guy right here, AUG, is the universal symbol that says start making protein here. AUG says start making protein here. So we read GCG, nothing. CGU, nothing. On and on and on. We get to AUG that says start making protein here. And it also says lay down the amino acid methionine. Every protein starts by having a methionine at the beginning. If they don't need it, it's cut off later. But methionine is at the start of every growing protein. And now we're going to read 3 letters at a time, 3 letters at a time, 3 letters at a time, until we get to one of these 3 guys that says stop making protein here. So here's our strand of messenger RNA. It leaves the nucleus, it attaches to a ribosome. Ribosome reads and reads and reads until it gets to AUG, and then it lays down methionine. Then it reads ACU, we look that up on our chart, and we get threonine. We read GGU, we look that up on our chart, and we get glycine, and we keep doing that. CGA is arginine. AUG in the middle, it doesn't say start because we're already going, it just says methionine. And we go hundreds or thousands of amino acids long until we get to one of those 3, the most common is UAA, one of those 3 that says stop. And immediately the ribosome falls apart. The ribosome falls apart and no more amino acids are laid down, even though there are going to be lots of mRNAs left. We have these amino acids all in a row, hundreds or thousands of them. Just a moment and we'll see what happens next. I left a space there because we have transfer RNA who's going to do a job. Remember, even though ACU says threonine, it's tRNA who goes and grabs the threonine and brings it back. How do we know which tRNA to use? Well, because the anticodon of the tRNA matches the codon of the mRNA. And anytime we have UGA as an anticodon for a tRNA, that tRNA that has UGA is carrying threonine. He brings the threonine along, he clicks together, he gives threonine to the ribosome who adds it to the methionine. Another transfer RNA brings glycine to the ribosome who adds it to the threonine, on and on and on. Why did I do the transfer RNA last? The order is DNA to mRNA. The tRNA comes along and hands the amino acids. There's a reason why I do it in the order that I showed you. You don't have to, but you're going to make a mistake if you do DNA, mRNA, tRNA, amino acid. See if you can decipher what that mistake might be. Well, if we look at our chart here, this chart is written in the code of RNA. It uses ACG and U. But what are we looking up on this chart? We're looking up messenger RNA. We look up ACU and it says threonine. If I put the tRNA here first, I might be really tempted to look up UGA. ACU says threonine, but if we look up UGA on the chart, where is UGA? UGA says stop, and that's not what we did. We didn't stop there. So it's best to do DNA, build our messenger RNA, look up the messenger RNA codons on the chart, build our amino acids, and then go back and see which tRNA we used. But now let's follow these amino acids. Here's that whole process that we just drew. Here's the DNA up here in the nucleus. Here we've unzipped part of it and made messenger RNA. The messenger RNA leaves the nucleus. When it leaves the nucleus, a ribosome forms. It starts reading along the DNA until it gets to AUG. AUG brings along a methionine, lays it down. Then we get to AUC. Then we get to the next one, The RNA keeps sliding along, the ribosome keeps sliding along the messenger RNA, 3 letters at a time. tRNA keeps bringing more and more and more amino acids. So the amino acid chain keeps getting longer and longer and longer until we get to stop. Then that growing chain of amino acid falls off. What do we call a chain of amino acids? A polypeptide. It's not a protein yet. It will be soon. So what happens? What's the point of this polypeptide in the protein? How do we get from polypeptide to protein? Let's take a look. We know that this polypeptide has a very specific order of amino acids. If it were a different strand of DNA, we'd get a different strand of messenger RNA, we'd get a different order of amino acids. So we have that order of amino acids. That is called the primary structure. The primary structure is the sequence of amino acids. One strand of DNA makes one sequence of amino acids, but a different strand of DNA would make a different sequence of amino acids. Remember the structure of an amino acid. We had a C in the middle. We had on one side an amine group. On the other side, we had carboxylic acid group. And coming off the middle, we had something called the reactive group, or the R group. Well, the R group is what determines the properties of that amino acid. And the R group is going to react with the cytoplasm. Some of them like water, some of them don't. Some of them are electrically charged, some of them are not. Some of them have positive charge, some of them have negative charge. All of them are going to react differently with the cytoplasm and with the neighboring amino acid. Based on how it reacts, it's going to fold up either into a coil called an alpha helix, or folded back and forth on itself, a beta pleated sheet. Sometimes it's just these long random swirls. But this is called the secondary structure. The secondary structure has these shapes based on the primary structure. If we had a different sequence of amino acids, the R groups would behave differently and we would get different alpha helices and beta pleated sheets and random coils. But once we have these, we'll have certain regions of that polypeptide that has alpha helices. Some regions will have beta pleated sheets. Some regions they'll be shorter or longer. Some regions will have coils. But based on which primary structure we have, we get a certain secondary structure. And once we have that secondary structure, the alpha helices, the beta pleated sheets, and the random coils will all begin to interact with each other. And they will push or pull and fold and twist, and ultimately they will take on a three-dimensional shape. Here we see a globular three-dimensional shape. Once we have a three-dimensional shape, this is the tertiary or third level structure of a protein. This is when it is a protein. The sequence of amino acids is just a primary structure. This is a polypeptide chain. They react with each other, we get secondary structures. This is just the secondary structure of a polypeptide chain. They react with each other, we fold up and we get a specific shape, a specific three-dimensional shape called the tertiary structure, and this is a protein. All proteins have primary, secondary, and tertiary structures. They are all based on the sequence of amino acids, which are all based on the order of DNA. Some proteins go a little bit further. Some proteins will have this that joins with another protein, and sometimes multiple other proteins. When that happens, and again, it doesn't always happen, but when that happens, we call that a quaternary structure. An example of this is your hemoglobin, the molecule in your red blood cells that carries oxygen. Hemoglobin has 4 pieces that stick together. But not all proteins have that. All proteins have tertiary structure. Some proteins have quaternary structure. But there's a lot going on there. If we look at that previous picture, there's a whole bunch of amino acids just growing and growing and growing. And this protein is going to fold up in a very specific shape, but a lot can go wrong there. Well, we have certain proteins called chaperone proteins, and it is their job to make sure that that amino acid chain, as it's growing, that it folds up the way that it's supposed to, because it could fold incorrectly. Chaperone proteins make sure that the growing chain of amino acids folds up correctly into the protein. Well, what happens if it doesn't? There's another protein that comes along called a proteasome. A proteasome takes a protein that is folded incorrectly, breaks the bonds between the tertiary structure or the secondary structure, lets it fold up again. It does that a few times. If it doesn't work, then it's just completely broken down into amino acids and we try to make the protein again. So chaperone proteins help with the folding. Proteasomes break the bonds so that we can try again or even dissolve the protein entirely. All right, that is the end of chapter 3. We are finished with chapter 3, the cell. The next thing we're going to do is jump towards the back of the book and look at metabolism. Specifically, we're going to look at cellular respiration, how our mitochondria make ATP. Okay? This is an extremely important chapter that we're wrapping up. If any of it doesn't make sense, ask me, ask your tutor, look at some other sources, whatever you need to do, but make sure to get those questions cleared up before moving on. I will say before you start the metabolism chapter, it's a really, really good idea to review from chapter 2 oxidation reduction reactions, redox reactions. We are going to have a very chemistry heavy chapter ahead of us. We cover very little bit of it, but that little bit that we cover has a lot of chemistry in it. No new chemistry. I promised you there would be no new chemistry. But the chemistry that we learned, we will be using. So make sure to review that and ask questions as we go forward before starting the next one. All right, I will see you all at the next lecture. Talk to you then.